histology education. For decades, the study of gut has been confined to the narrow, singular view of traditional microscopes. Today, we are changing that experience entirely. Through today's session, Visualizing the Gut, GIT Histology through Digital Microscopy, we will explore how digital microscopy is transforming the way histology is taught, learned, and understood. We are moving beyond the limitations of crowding around a microscope. Now, before we proceed, just a gentle reminder to all our participants to ensure that they drop in their full name in the chat in order to receive participation certificates post the session. We are honored to have with us Dr. Tejasvi Lokanathan, Professor and Head of the Department of Anatomy at Adi Chinchanagiri Institute of Medical Sciences. Dr. Lokanathan isn't just speaking to us today. He is giving us a front row seat to a live digital exploration of GIT histology using a silica microscope. He will demonstrate the art of seamless tissue mapping in real time, navigating across high-resolution slides to help us identify and understand the intricate layers of the gastrointestinal tract. Throughout the session, if you have any questions, please drop them in the chat and we'll be addressing them at the end. Dr. Lokanathan, thank you so much for joining us to share your expertise on this. We are eager to hear your insights. Over to you.
Thank you, ma'am. Thank you for that kind introduction. If you could please confirm, like the screen is visible, the slide.
Yes, it is, sir. It is visible. You can go ahead.
Before I start, my sincere thanks to MedPrime for giving me an opportunity to present this webinar. Thank you very much for hosting me. So what we'll do today is we'll try to navigate through all the slides of GIT, starting from the slide of esophagus till the slide of colon, restricting the histology discussion to the slides of the alimentary canal. For time being, we'll not discuss the glands associated with the GIT. Our main focus is on identifying different parts of the GIT and to understand how to differentiate each of these parts of the GIT under microscope. We will be using HND stained slides, which are often used in UG exams across various universities in the country. And we will stick to 10x magnification unless I shift it to a higher magnification to show a particular structure. As ma'am rightly said, if you have any questions, please drop it in the chat box. We would try to address it by the time we close this session. So let's get started with the slides of the GIT. And I will be using the silica microscope with a digital head to demonstrate the slides live. So what we have currently is the slide of esophagus, which is the first or the proximal part of the GIT. The first question is, how do we identify this as a slide of GIT and then as a slide of esophagus? Let's try and do it now. The first thing we need to identify is the 4 distinct layers of the GIT. As we know, the GIT is made up of 4 layers. This entire extent is known as the mucosa. Then we have the submucosal layer. Then we have the muscularis externa. And then we have the adventitial layer. Whenever part of the GIT is covered by peritoneum, we would change it as serosa. Basically, a layer of mesothelium along with subserosal connective tissue. Right? You take any part of the GIT, the organization is the same. 4 layers. It is how these 4 layers differ in each part of the GIT that will help us differentiate between the various parts of the GIT. Right? For example, when we talk about esophagus, the function of esophagus is to allow the transport of food from the stomach from the pharynx to the stomach. Correct? So basically it acts as a conducting pathway. Since food passes directly through the esophagus, the mucosa is modified to withstand the wear and tear. So let's try to identify the different layers of the mucosa now. The first layer of the mucosa itself, as the pointer is showing, this is the lining epithelium of the esophagus. The lining epithelium is deep to the lining epithelium, we have the layer of lamina propria. The muscularis mucosa, which should separate the mucosa and submucosa, is not very distinct here. As we move to stomach and rest of the parts of the GIT, this will become very well defined until we reach the slide of colon or vermiform appendix. Right? Now, if we observe the lining epithelium, the lining epithelium appears to be multi-layered or stratified. The functional significance of this is esophagus has to withstand the passage of rough food particles through its lumen. So it cannot have a simple epithelium, and that is why this has the stratified epithelium, which is stratified squamous non-keratinized. As I said, this is how it looks under 10x magnification. If I just shift it to the higher magnification, that is 40x, let's try to take a look at the lining epithelium. We can see the wonderful appearance of stratified squamous non-keratinized epithelium. When we identify the slide of the GIT, this is the only slide of the GIT which is actually lined by stratified squamous non-keratinized epithelium, unless we are looking at the terminal portion of the anal canal or the oral cavity. Right? So this is how we confirm that we are looking at the slide of esophagus. Look at the mucosa, lining epithelium is stratified squamous non-keratinized, and that will confirm that we are looking at the slide of esophagus. Let's shift back to 10x magnification and try to identify the other layers of the esophagus. Deep to the lining epithelium, this is the layer of lamina propria. Lamina propria is basically made up of loose connective tissue which is richly cellular, and most often the cells are made up of lymphocytes. Right? This is limited from the submucosa by a thin layer of muscularis mucosa, which is not clearly or distinctly visible here. As we see the slide of the stomach, we'll try to identify this. Then, when we come to the submucosa, we can see a lot of esophageal glands. These are basically mucus secreting glands which will allow the lubrication of food particles that pass through. Right? These submucosal esophageal glands are present in the dense connective tissue. Lamina propria, loose connective tissue. When we talk about the submucosa, it is made up of dense connective tissue. When we come further deeper, we can see a distinct layer of the muscular external or muscularis externa, which seems to be arranged in two layers, the inner circular and the outer longitudinal. You might ask, the arrangement looks the same in both the inner layer and the outer layer, they are appearing circular. This confusion is because the way the section has been taken. If the section has gone little bit oblique, we will not be able to identify the longitudinally arranged smooth muscle layer. Right? So what we have here, the inner layer would be described as inner circular and the outer one would be outer longitudinal. Peculiarity of the esophagus is the upper one third of the esophagus, the muscularis externa, is made up of entirely skeletal muscle, which is an example for visceral striated muscle. When we come to the middle one third, it is a mixture of both striated and non-striated, that is skeletal and smooth muscle. When we come to the distal one third, this is purely made up of smooth muscle. So based on the type of muscle present in the muscularis externa, we can tell whether this is taken from the upper one third or the lower one third or the middle one third of the esophagus. Right? Done. Then if we come to the last layer, this is the layer of adventitia, which is made up of connective tissue again, and this allows for passage of larger blood vessels and nerves. In fact, we can see section of one peripheral nerve here and one blood vessel here filled with adipose tissue. So this entire layer is called as adventitia. Only the intra-abdominal part of esophagus will contain serosa. So remember the thumb rule. If a GIT organ is covered by peritoneum, it will have serosa. If there is no peritoneum, then it will be purely adventitia. In addition to all these features, when we take a look at the muscularis externa, between the circular and the outer longitudinal layer, we invariably get a nerve plexus. We will try to identify one such plexus a little bit later, and that is what we refer to as the myenteric plexus or Orbach plexus. This is responsible for maintaining the peristalsis. Similar to this myenteric plexus, when we talk about the submucosa, within the submucosa, we will get Meissner plexus. We will try to identify these two nerve plexus as we move from esophagus to the vermiform appendix. This is a brief description of how to identify and how to explain the slide of esophagus. This would be particularly useful for all the first year medical students who are attending this session. Right? Your practical exam will contain an exclusive discussion session on general histology slides and systemic histology slide, where the examiner would spend at least 3 to 4 minutes discussing the slide. And this is how you should explain it. Right? Done. Now, once we are done with the slide of the esophagus, let's move on to the slide of the stomach. Stomach, as we know, is divided into different parts. Histologically, it is divided into three main parts. That is cardiac, close to the esophagus. Then we have the fundic region, which includes majority of the stomach, and then the pyloric region close to the duodenum. Now, how does it look? Let's try to understand. We'll try to focus starting from the mucosa again, and then we'll try to differentiate this from the slide of esophagus. Now, this is how the slide of stomach will look. Very often, where the students falter, especially in GI sliders, to differentiate between various parts of the stomach and the small intestine. Now, how do I say that this is the slide of stomach? This is the first question every examiner will ask. How did you say that this is the slide of stomach? Your answer should be, sir, I can see 4 layers of GIT. This is the mucosa. Then we have the submucosa. Then if we move little bit further, we can see muscularis externa. And now we have the last layer, that is serosa. So stomach will contain serosa, not adventitia. Now the next question is, how do I say that this is the slide of the fundus and not the pylorus of the stomach? The mucosa here contains small gastric pits, or we can say shallow gastric pits. You can identify these, right? We will just try to write it down. This is one gastric pit, this is one more gastric pit, and this is one more gastric pit. Opening into the gastric pit, we have the entire gastric gland, which looks basophilic in the basal portion and eosinophilic in the middle. This is because of presence of specialized cells in the fundic glands. So mucosa shows shallow gastric pits lined by simple columnar epithelium. Opening into the bottom of the pits are gastric glands. Now, gastric glands contains two major types of cells which can be clearly identified by shifting it to 40x magnification, which we'll do now. So I'm shifting the magnification to slightly higher magnification now to show you two important cells that actually make up the fundic glands. Let's see if you can identify them. This is a beautiful demonstration of two major types of cells that make up the fundic glands. For example, can we make out these eosinophilic cells? All these eosinophilic cells, these are the parietal cells which are responsible for secretion of hydrochloric acid. These are typically pear-shaped. They are identified by intense eosinophilic staining. Whereas, if you compare that with these cells, These cells are the chief cells which secrete pepsinogen. Right? Now, these chief cells appear basophilic because of rich, rough endoplasmic reticulum. The apical portion contains zymogen granules which appears eosinophilic, but the basophilia overtakes it. If your examiner asks, how do you differentiate between parietal cell and chief cell? This is how you should shift it to high par and show it. Right? The Fundy glands has these two main types of cells. Apart from this, there are neuroendocrine cells which are not visible. If we move further higher up, right? Let's remove these markings. What we have here, these are called as the surface mucus cells. As we come close to the glands, here we have the mucus neck cells. These two types of cells produce different types of mucus. The surface mucus cells produce an insoluble mucus, whereas the mucus neck cell produce soluble mucus. This is the main difference. Done. So we are discussing about the mucosa where we described shallow gastric pits and cells of the gastric glands. Let's shift back to 10x magnification, which you often use in your exam, and let's try to identify the next layers. If we go further, the immediate layer we need to identify is the lamina propria, which extends between the glands. Separating the submucosa and the mucosa, can you see this distinct muscular layer? This is what I mentioned as muscularis mucosa. Even muscularis mucosa is made up of two distinct layers, circular and longitudinal. Right? Muscularis mucosa function, this separates the mucosa and submucosa. The submucosa, as we can see, is made up of dense connective tissue with few blood vessels. The peculiar feature of the stomach is it has a thick muscular wall. Now, can you see these nerve cells in between the muscular layer? This is what I mentioned by the mesenteric plexus or Auerbach plexus. Right? Now, what we have in the muscularis externa is three distinct layers: inner oblique, middle circular, and outer longitudinal. And this is a peculiar feature of the histology of stomach. Instead of two-layered arrangement, we have three-layered arrangement. But again, this is very difficult to identify. But for your exam purpose, you can mention these three layers. Right? Then, if we come deeper, as the pointer is suggesting, this is the thin layer of peritoneum, which is the serosa. Serosa also contains the thin subserosal connective tissue, but the mesothelium forms the lining or the covering. This is how you should describe the structure of the fundus of the stomach. Now, the difference between the fundus and the pylorus will be striking. What I want each one of you to do is focus on the depth of the gastric pits, and the difference between these two will be clear, right? So what we'll do now is we'll try to focus the fundus of the stomach, right? So here we have, the pylorus of the stomach. So let's see if we can focus the mucosa, right? I'll probably try and focus one particular layer where the gastric pits are a lot deeper so that you can get a clear understanding of this. Fine. So take a look at the gastric pits here. The gastric pits appear much deeper, right? So take a look at these gastric pits. The gastric pits are much deeper in the pylorus, and the characteristic appearance of the Fundy glands is lost. The pylori glands differ completely from that of the Fundy glands, which contain plenty of parietal cells and the chief cells. So deeper gastric pits and pylori glands. This will help us differentiate between the fundus of the stomach and the pylorus of the stomach. If we come to the submucosa is unremarkable, right? When we talk about duodenum, the submucosa contain a peculiar feature called as Brunner's glands. Otherwise, submucosa is unremarkable in stomach, in jejunum, right? If we go further deeper, we have the layers of the muscularis externa, and lastly, we have the serosa. Let's see if we can identify the meyenteric plexus. Can you see this here? I'll just mark it for you again. This is what we call as meyenteric plexus, which contains nerve cells of the autonomic nervous system. Right? Now, these are responsible for maintaining peristalsis of the gut. We can shift it to high power and confirm, are we looking at these nerve plexus? Right? Let me remove this and let me focus it for you. Right? So what we have here, this is what we mean by the nerve plexus or the meyenteric plexus. These are present between the inner circular and the outer longitudinal layer. Right? So this is how we should differentiate between the two parts of the stomach. Right? So what is the giveaway clue for you to identify the pylorus? Take a look at the gastric pits. Shallow gastric pits, fundus. Deep gastric pits, it is pylorus. This is how we need to identify. Done? Right. Let's move on and identify the rest of the slides. Once we are done with the slides of stomach and the esophagus, now we have the slides of small intestine, and this begins with the slide of duodenum. Right? Now let's see how the slide of duodenum looks under 10x magnification. Right? So this is the slide of duodenum. Now again, the first question is, sir, how do I say that this is the slide of duodenum? When we talk about the slide of small intestine slides will have what we call as villi. Right? Now let's try to understand what exactly are villi. Right? Now, when we have the surface epithelium, let's imagine this is the basement membrane. If the surface epithelium invaginates, it gives rise to formation of the gastric pits, gastric glands or intestinal glands. But when it projects above the surface, it gives rise to what we refer to as villi. Villi are basically finger-like projections. Let me repeat again. These are finger-like projections, whereas crypts or intestinal glands, these are invaginations. They invaginate into the lamina propria, whereas lamina propria will extend into the core of the villi. The first question, what are these villi and why do we have villi? Small intestine, the major function is absorption of nutrients. The appearance of mucosal folds, villi, microvilli, all these will increase the surface area of absorption, and that is why we have villi in small intestine. So remember, if you see a projection above the lining epithelium, it has to be the slide of small intestine. You need to differentiate whether it is duodenum, jejunum or ileum. Now, let's go back to the slide and try to identify why did we call this as duodenum. Now, this is the first question the examiner asks and your answer should be, sir, again, I can identify 4 distinct layers of GIT. What are they? This entire layer is mucosa. Then we have the submucosa, we have the muscularis externa, and finally we have the serosa. The mucosa contains finger-like projections which are called as villi, the core of which is formed by lamina propria. This is the layer or this is the line of surface epithelium. Projecting from this is one single villi and this is one more villi. So core of the villus here is formed by what? lamina propria, which contains plenty of lymphocytes here, right? Then, if you come lower down, can you see this thin eosinophilic layer? What is this layer? Can you tell me? This is the layer of muscularis mucosa. Are we clear? Now, muscularis mucosa again is separating the submucosa from the mucosal layer. Correct? So, broad villi, right? Club-shaped villi with the submucosal Brunner's glands. This is the characteristic feature of the duodenum, right? If you scan through the entire the submucosa contain, can you see this? These are the Brunner's glands, right? Brunner's glands secrete mucus again, which will neutralize the acid. Done. Now, these Brunner's glands are the characteristic feature of duodenum. These are present in submucosa. When we compare this with the slide of jejunum and ileum, we will understand a clear difference. Done? Right. If we move further down, can you identify this? What is this layer? Now, this, just a minute. Yes. This is the layer of the muscularis externum. Again, inner circular and outer longitudinal layer. Again, in between, you will get mesenteric plexus and all that. And once we come deep down, the last layer would be the serosa. Remember, except for the proximal 1 centimeter of duodenum, rest of the duodenum is retroperitoneal. So the first 1 centimeter or 1 inch of the duodenum has serosa all around. Rest of the duodenum will have serosa on only one side. The rest of it is covered by the adventitia. What is the importance of this? If you are a pathologist looking at the specimen of duodenum and wondering why there is no peritoneum, why there is no serosa, possibly the section has been taken from the opposite side of the retroperitoneal part of the duodenum. This important difference we need to identify. So how do you summarize this slide to the examiner? Sir, this is the slide of the duodenum. I can make out 4 distinct layers of the GIT. The layer of mucosa, submucosa, muscularis externa and serosa. The mucosa is made up of 3 layers: lining epithelium, lamina propria and then muscularis mucosa. The lining epithelium projects as broad villi, the core of which is formed by lamina propria. The muscularis mucosa separates mucosa and submucosa. The characteristic feature of duodenum is the submucosal Brunner's glands. Then we have the 2 layers of muscularis externa and the serosa. You will have 2 to 3 minutes to do this in front of your examiner, and whatever structure he asks you to show, you need to show them. And this is how we need to identify the slide of duodenum. Done. Now, let's try and compare this with the slide of, possibly I'll just make a small change now and let me ask you if any of you can identify this particular slide. Try and identify this slide because you would have already seen these slides in your regular classes. Now try and identify what is this slide. Anyone? Any one of you just try and identify what is this slide that we are looking at. Is this the slide of esophagus, stomach, small intestine, large intestine? What this slide could be? Okay, Virendra says jejunum, Varsha says ileum. We'll try and differentiate if it is the slide of jejunum as Virendra says, or as Varsha and Sunavya says, is this a slide of ileum? Yes, Virendra, you got it correct now. This is the slide of ileum. Now, why do I say that this is the slide of ileum? Take a look at the submucosa, right? The entire submucosa as well as lamina propria is studded with lymphatic follicles, which we refer to as Peyer's patches. If we take a look at the villi appear leaf-shaped. This is the characteristic term that you should use. Villi in case of ileum are broad and leaf-shaped, and the lamina propria, right? The lamina propria contains the lymphatic cells or the lymphocytes. These lymphocytes extend into the submucosa. This aggregation of lymphocytes or lymphatic nodule is what we call as Peyer's patch, a peculiar part of mucosa associated lymphoid tissue responsible for the gut health or gut immunity, right? So broad leaf shaped villi with Peyer's patches. The diagnosis is the slide of ileum. The rest of the features are unremarkable. We have the muscularis externa and we have the layer of serosa. Done. Remember, when we talk about jejunum or ileum, it is completely covered by serosa. There is no question of adventitia because the entire length of the jejunum and ileum is intraperitoneal. Right? So this is the slide of ileum. Now let's see how the slide of the jejunum will look. Right? So let's focus the slide of jejunum and let's see if we can identify this. So here we have the slide of jejunum. Now the first question is, how do I differentiate this from the slide of duodenum or the ileum? Let me show you one peculiar feature of the jejunum, and that is this region. Can somebody identify, right? Can somebody identify this particular region for me? What do we call this region as? Anyone? This is the slide of jejunum, Revati, and I want you to identify this particular layer. What exactly is this region known as? This region, yes, this is submucosa, right? Now, let's take a look at, yes, it is submucosa, I totally agree. We can identify the villi here, right? The villi here, if you take a look, they are long and finger-like. Okay, so long finger-like villi are the characteristic feature of jejunum. But if we see further down, what we see here is a submucosal fold. This is what we call as the valves of Kerkring. Stomach has rugae, which are temporary mucous folds, whereas small intestine has the mucous folds, crescentic mucous folds, which are permanent mucous folds, and these are called as valves of Kerkring, right? The core of which is formed by submucosa. Here there is muscularis mucosa, and then if we go above, we have the we have the intestinal glands, and then the villi. Right? So this is the characteristic appearance of jejunum. Jejunum has longest villi of all the three parts of the small intestine. The core of the villi is again formed by lamina propria. These are the intestinal glands, and then we have the muscularis mucosa. Deep to the muscularis mucosa, we have what is this layer? This is the submucosa. So if we see slightly further in this region, the submucosa, remember, is unremarkable. In case of duodenum, this had the Brunner's glands. In case of ileum, this had Peyer's patches, whereas in jejunum, the submucosa is unremarkable. You can say no important structure is present here apart from blood vessels or the nerves. This is how we should differentiate or identify the slides of jejunum. Invariably, when they focus this slide for your exam, they will focus a mucosal fold and they will focus this region, that is the entire villi on the top of mucous folds. Let me shift it to a low power objective so that you can identify this much more clearly, right? This is the 4x objective, and what I wanted to show you was this region. Okay. Now, if you observe, this is the mucosal fold or the valve of Kerkring, on top of which you have finger-shaped villi. If the examiner decides to focus this under 4x, you will get this view. If it decides to focus it under 10x, which is most likely possibility, this is the region that they will tend to focus, so that you can identify both villi and the crescentic mucous folds. Are we clear? So this is how we should identify the slide of jejunum. Done? Right. Now, once we are done with the slides of the small intestine, let's move on to the large intestine. Right? The large intestine, the proximal portion, let's say we have cecum and appendix, then we have colon. First, we'll see the feature of colon, and then we will try and shift to the rest of the parts of the large intestine, that is the vermiform appendix. Now, usually this is the field that the examiner will focus. Where many students falter again is to differentiate between a villi and an intestinal gland. Remember, villi are present only in small intestine, duodenum, jejunum and ileum. They are present nowhere else. Stomach, you have gastric pits, and then you have gastric glands. When we talk about large intestine, what we get is these crypts of Lieberkühn. These are the simple tubular intestinal glands. What is the most characteristic feature? If you take a look at the cells lining the intestinal crypt, they are typically what type of cells? Goblet cells. What is a goblet cell? If you remember, goblet usually refers to a wine glass which has a broad apex or a top portion and a narrow base. This is the characteristic feature of a goblet cell. Since it appears a goblet, the mucous cells are called as goblet cells. I want you to see this under high power, how these look. This appearance you can never forget. Can you see this? All these are your goblet cells. Right? Now, goblet cells, why they appear like this? This is one question you will be asked in exam. The apical portion of the goblet cell, remember, will have a large mucin granule. And this mucin granule gets washed off during the process of H&E staining. During H&E staining, we use a reagent called as xylol, which dissolves the fat or the mucin. Now, once the fat or the mucin dissolves, there is nothing to stain. Once there is nothing to stain, the whole area will appear empty, which is why we have the goblet appearance for mucus cells. If you take a look starting from the stomach to the large intestine, the number of goblet cells will increase. Right? So, how do we describe the structure of the large intestine? This is the slide of colon or large intestine. We can identify the entire layer of mucosa limited by the muscularis mucosa. Then there is submucosa. Then there is muscularis externa. You can make out the two layers classically here. This is the inner circular layer and this is the outer longitudinal layer. And then of course we have the thin layer of serosa. Right? The mucosa contains simple tubular glands arranged in the form of intestinal crypts or crypts of Lieberkühn, which are largely lined by goblet cells. Then the thin lamina propria is preceded by thin muscularis mucosa. Then we have the dense connective tissue, the submucosa. Then the two layers of muscularis externa. Even in this, you can make out beautifully arranged myenteric plexus or Auerbach plexus. Now, these are the nerve or the ganglion cells of myenteric plexus which separate the circular layer and the longitudinal layer. Right? This is how we should identify the slide of colon. One more important feature of colon is the longitudinal layer will be thickened to form what we call as tinea coli. And let me demonstrate how the tinea coli actually looks. Right? We will use a different slide to identify the tinea coli. This is again the slide of, let me focus it this way. So this is the slide of the large intestine. Again, we can see the epithelium and the mucosa. If you take a look, this whole layer, right? This whole layer is the inner circular layer. This layer is the longitudinal layer. So let's try and move this and see if we have aggregation of these longitudinal bundle. There is no tinea coli on this side. Let's try to identify the opposite side. Can you take a look here? We have the thick longitudinal arrangement of the longitudinally arranged thick bundle of muscle fibers, and this is how your tinea coli will look. This is the circular layer, this is the longitudinal layer, this is the thick tinea coli. If this is there in your slide, make sure that you show this to your examiner when the examiner approaches you for your discussion. Right? What are tinea coli? Tinea coli are aggregations of the longitudinal smooth muscle bundle. Done? Right. So this is the slide of colon. The last slide in the GI tract is the slide of appendix, and let's see how this looks under microscope. So here we have the slide of the vermiform appendix. Now, what is the characteristic feature of vermiform appendix? Vermiform appendix will neither have the intestinal glands nor the villi. The intestinal glands here are rudimentary, and these are what we call as rudimentary crypts, right? So again, we can identify the mucosal layer. Then we have the submucosa, we have the muscularis externa, right? One more feature I just want you to appreciate here is uh, take a look at this part of the appendix. Can you make out this thin layer of muscularis mucosa? This thin muscularis mucosa or disrupted muscularis mucosa is a characteristic feature of the vermiform appendix, right? We can identify the lymphatic nodules in the lamina propria and submucosa. We can identify rudimentary crypts, again lined by plenty of goblet cells, but disrupted muscularis mucosa is one feature the examiner will expect you to answer, right? Even if you talk about all the four layers, describe all the four layers, disrupted muscularis mucosa is one key answer that the examiners always expect from a student, right? And this you should highlight, right? So this is how the slide of vermiform appendix will look, right? Now, what I've tried to do in last 45 minutes is to explain all the slides of GIT under 5 minutes so that you get a comprehensive understanding of how to explain these slides in exams, right? So my simple tip, start with identifying all the four layers, tell the examiner what slide are you looking at, and then describe and demonstrate the peculiar feature of that part of the GIT. For example, if you are looking at the slide of duodenum, show the examiner Brunner's gland first and then show everything else, right? So Brunner's gland is the characteristic feature of duodenum. The Peyer's patches are the characteristic feature of ileum. So show these first and then try and show rest of the layers. If the examiner asks any peculiar feature to be demonstrated, show it under 40x. Unless told, never ever use high power magnification in your exam. You will end up breaking the slide and probably your whole exam will again change, right? So never ever use high magnification unless it is asked by your examiner. Done? Now, let's see if you have understood what we have discussed till now. Let me focus few slides randomly and you will identify what slide am I focusing. Okay. So here is the first slide. Tell me what slide this is. I don't want any description. Just type in the chat box what is the slide that you are looking at. Right? So take a look at the slide and tell me what is this slide that you are looking at. Just the name of the slide. We did discuss about 9 slides as of now. Let's see if you can identify this slide. What is this slide? I will just move this back and forth so that probably you'll get a clear understanding of what we are looking at. Fine. Excellent. Tushita, Dhruva, Aishwarya, yes, it is the slide of pylorus. Excellent. Done. Now we will take this out, right? Let me focus one slide now, and let's see if you can identify this. So can you identify this slide? Yes, this is the slide of large intestine. Excellent. Now remember, I did not focus the same area that I showed you in the beginning. This is what each one of you must be able to do, right? If the examiner just puts this on the screen, you should be able to tell what slide this is. Why I'm telling you this is the examiner may not focus the same field that your internal examiners might have trained you with, right? So you need to scan the whole slide. In Spotter, the examiner will definitely focus the most relevant feature so that you can easily identify. Otherwise, when it is given for discussion, you should be able to identify this slide very clearly. Very good. Excellent. So this is a slide of colon. Agreed. Right? Now let me focus this slide and let's see if you can get this correct. Fine. So identify this. What slide could be this? Again, what I'm trying to do is I'm trying to focus the most peculiar feature of this particular tissue. Excellent. This is the slide of duodenum. How do you look at duodenum? Again, I'll tell you, take a look at the submucosa. If you identify a slide as small intestine slide, the first thing you need to look at is look at the submucosa, nothing else. If submucosa has glands, it is duodenum. If submucosa has lymphoid tissue, it is ileum, right? If there is nothing and there is long finger-shaped villi, that is the slide of duodenum, right? So, I'm sorry, jejunum, okay? So let me focus this now and let's see if you can get it correct. Fine. See if you can get this correct, or I'll focus maybe another region as well. Now this exactly is the slide of jejunum. Now this is what I want each one of you to do, right? Make sure that you identify the slide by feature, not by the identification points on the slide. The slide is broken, the edge is broken. This should not be the identification point. Identify the tissue and come to correct diagnosis. Okay, let me focus one last slide for you and let's see if you can identify this correctly. Okay, again, I'm telling you, I'm not focusing the field that I described. I'm focusing a different field altogether. So can you identify this? What is this slide? I will just uh, keep it moving so that you can identify other regions as well. Okay, fine. So this exactly is the slide of fundus. Now let me check if you have understood what we discussed in the beginning as well. I will focus a particular cell now and I want you to identify what cell it is. Okay. So can you tell me what cell? Sorry. Can you tell me what cell is this which I have focused? Excellent. That is the parietal cell. And what cells are these? These are the chief cells. Right? Now, this is the extent to which you must be able to identify the different types of cells present in a particular tissue. Right? So I'll tell you again, the difficulty lies in differentiating between the two parts of the stomach, differentiating between the three slides of small intestine. If you take a look at these slides together, whenever revision happens in your college, try and take a look at these slides together, compare these slides, and this will make it very clear for you how to identify this. Done. This is what I had to explain or to all of you. I thank each one of you for joining this. Now we will take up questions if any. I request ma'am, if there are any questions in the chat box, let me know so that I'll try to answer these questions before we close.
Yeah, so I don't think we have received any questions so far. We can just give them a few minutes.
Sure.
If there are any questions, please drop them in the chat box.
In fact, ma'am, like most of the students I see are my own students, and recently their performance in histology has like really gone up, and we are really happy with like how they are doing. And we consistently use silica for demonstration. All our faculty use the microscope where they demonstrate the features. The students now themselves they do a seminar where they focus the slides and they identify them. So we have got a good bunch of students with us, and I'm really happy that like most of them have joined here today for this webinar.
That's
In,
really good to hear, actually. And that's exactly, what we had in mind when we were developing the product, that, this is how education and teaching should be. It should be this easy, right? Because I remember I am a biotech student, so I remember the way it used to be in my days, and, using the microscope was like a very terrifying thing. And it really makes me happy to see how things have changed now and how easy it is to, take classes that 200 students are able to see the same thing at the same time, sitting wherever they are. So I mean, really happy to see this and to hear it from you as well.
one more thing we did, ma'am, is one of their exam station is what we call as spotters, where we usually keep 10 different microscopes. We focus slides, and then we ask students to rotate among these 10 stations, identifying the slide. What used to happen was like if one student just touches the objective, the field would totally change, and most of the students would falter there identifying what slide that is, not because of their fault, because of someone else's fault, right? So what we have done using Silica is even for our university exams, our spotter exams happen on the screen, where we focus, the examiner focuses the slide so that he has that freedom to show what particular region will give the students the diagnosis, and 25 students at once, without moving, without rotating, can actually answer at the same time, right? So it has given us like a lot of freedom, and as I told, uh, performance of our students have really improved over like last few months as our faculty give feedback. The way they get to identify the structures on a bigger screen or using an iPad is different from what they would look at traditional IPs. I'm still waiting for questions. Possibly, I don't know. The students are like not coming out with questions. It's okay.
I think it speaks to, the depth of coverage that you have provided on the topic, that there are no questions. I think you've covered, everything pretty well. That's probably why there are no questions.
Yeah.
Um,
I hope so,
but
ma'am. Hope so that like they have understood and
yeah,
we,
Revati is agreeing with me.
yeah. Right. So, as I said, really proud of these kids, ma'am. They're being pretty good now. I know that like they are under a lot of pressure with their internals and exams coming up, and we keep pushing them regarding like their studies, but still they come on top. So, really glad that like all the kids joined and made this a good session. I once again thank MedPrime, and you have got a wonderful product. It's doing like really wonders in our institution. Thank you again.
Thank you, and really excited to hear this feedback from you, and we are hoping to make this a regular thing in the future as well. Um, so I think if there are no questions, we can wind up today's session. We want to thank you for this masterclass. I mean, it has been a departure from the regular traditional static way of learning, and we hope that this is the path forward in medical education. And to all our attendees, thank you so much for your time and engagement today. We hope you found this session extremely useful. As a quick final reminder, please keep your eye on your email in the next few days for your participation certificates. And do follow MedPrime on our social media platforms to stay in the loop for our upcoming episodes. We have many more groundbreaking topics on the horizon that you won't want to miss. So, on behalf of the MedPrime team, thank you so much for your curiosity and your time. Have a wonderful evening, and we look forward to seeing you at our next webinar. Thank you, and have a nice evening.
Thank you again, ma'am. I once again thank all my students for joining this session and making this a success. Right? Thank you again, ma'am.
Thank you. Thank you, everyone. Take care.